Hello, could somebody confirm that you're hearing me?
I can hear you.
Great, thank you. Let me go ahead and share my screen. I think we're ready to begin. My voice sounds a little hoarse today, so please bear with me. So today is November 8th, and we're supposed to be working on microbial genetics. Hopefully we'll finish this. I doubt we will. We're a little behind schedule. There is no class this Thursday because of Veterans Day, and Veterans Day is really on Friday, but I put it in the schedule that we would take Thursday off, and I don't know if that's something the school is doing or not, but last year we had Veterans Day off, and so I just built it on last year's calendar. There is a lab today at 6:30. And that'll be lab 9. Any questions about any of this? There is no quiz this week. All right, that's it there. Let's go ahead and go to chapter 8.
I did have a question about next week's quiz.
About next week?
Yeah, the quiz for next week.
Uh-huh.
It says 0.24 points. Is there only one part, the respondus lockdown?
There is only one part on quiz 5. I think it's quiz 5B, but it is the multiple choice questions, yes.
Okay,
It's not worth whatever value it says it is. I have it as that in the computer because you get to drop out one quiz. And if you were to get points on quiz 5, then you would have, do I still have my syllabus here? Then you would have too many points for the quizzes, and you would have too many points. You'd have more than 720 points. You would have, let's see, 375 points in quizzes, and the quizzes are only supposed to be worth 300 points because you can drop your lowest quiz. So quiz 5 will be worth 75 points. I have it in the Canvas as close to 0 so that the number of points in the quiz are correct. And what I'll do is I'll manually go in and check the quizzes and drop the lowest quiz. That'll make your total quiz points worth 300 points. And then for quiz 5, I will go in and bump up the points up to 75. And all of that will be done manually.
okay. Do you go
Okay.
through the multiple choice questions by chance?
What do you mean?
Like, do you go through and see, like, change the grade at all if it's
I'm not sure I understand. What I'll do is the system in automatic quiz, it will grade it itself, and I will just change the value from 1 point or 0.1 point, something like that, to whatever it's supposed to be. And so the total points will be changed from, was it 0.24 points? It'll
yeah.
be changed from that to up to 75 points.
Okay. All right,
Okay.
thank you.
And I thought I had that discussed in the discussion of that quiz. All right, let me see, did I ever open up? I did not. Well, that was the last one we did, so I can find it. So I believe we were talking about operons, is that correct? All right, I haven't heard anything, so I'm going to assume we were talking about operons, and we talked about the lac operon, and how the polymerase will bind to the promoter and then transcribe the DNA moving down. And the lac operon is an inducible operon. It's normally turned off. I believe I mentioned all of that. Inducible means it's normally turned off, and it's normally turned off because E. coli is not normally around the sugar lactose. You only would find the sugar lactose if you're really on a dairy farm or around dairy. And that's why E. coli normally has the lac operon turned off, because it doesn't normally see the sugar lactose. So why bother making the enzymes you need for a sugar that you're not going to most likely use? How the lac operon is turned off is there's an inducible gene. This is another gene, and it's not a part of the lac operon. It has its own operon. And this gene is a regulatory gene that makes a repressor protein. The repressor protein will come and bind to the operator. That is the region here, with the O, it's not marked there. I didn't notice that. Which is the region immediately downstream of the promoter site. So now when the RNA polymerase binds to the promoter and moves down, the RNA polymerase gets blocked by the repressor protein. And it can't transcribe the genes of interest, which are the genes for lactose and the transport protein to bring the sugar lactose across the cell membrane, and then the other gene involved in the metabolism of lactose. So that is how the gene is turned off. Another gene shuts it down. Normally, the genes that regulate an operon are nowhere near the gene that they regulate. In eukaryotes, most of the times, the regulatory genes are in a different chromosome than the gene that they regulate. In the case of the lac operon, the regulatory gene is right next to the lac operon. Any question about any of that? All right. So how does the lac operon get turned on? The lac operon gets turned on when the sugar allolactose is around. Allolactose is a stereoisomer of lactose. So whenever you have lactose around, you'll have allolactose around. Any questions about that? Meaning one is simply the folding of the sugar in the L form and the other in the D form, and I don't remember which one is which. It's not terribly important. The point is that whenever the sugar lactose is around, the sugar allolactose will be around. Allolactose is the inducer. It will bind to the repressor protein and inactivate the repressor protein. When the repressor protein is inactivated, it cannot bind to the operator. Okay? And then the lac operon will be turned on because the repressor protein can't turn it off. What happens in this situation? What happens to the repressor protein that's bound to the operator already? Anyone have a guess? Well, there's really only two possibilities. All right, the first possibility is that, and this is, well, actually the first possibility, I guess we'll say, is not what happens, and that is the repressor protein gets bound to allolactose, and then that inactivates the repressor protein. That actually doesn't happen. When the repressor protein is bound to the operator, allolactose cannot bind to it. What happens is the repressor protein falls off of the operator by a stoichiomic reaction, meaning every once in a while, the repressor protein falls off. It's not a problem because if there's other repressor protein around, one of these repressor proteins or the one that just fell off will rebind to the operator, shutting down the gene. However, in the case where the allolactose, the inducer, is around, once the repressor protein falls off, allolactose will then bind to it and then inactivate the repressor protein. This will mean the lac operon is turned on, and then the polymerase will bind to the promoter and then transcribe the DNA, making the messenger RNA for the genes of the structural genes of the lac operon. Any question about any of that? What do you notice that's unusual about this messenger RNA? Come on, somebody should notice it. How many proteins do we get made from that messenger RNA? All right, that is unusual once you answer the question. And that's what we see in prokaryote operons, that occasionally there can be more than one protein made from one messenger RNA. And it just depends on how many structural genes are in the operon. On a eukaryotic operon, you only have one structural gene in each operon. And what do we do?
Do the proteins break apart in prokaryotes afterwards, or do they stay in that 3-protein configuration?
No, they become isolated proteins.
Oh, okay, thank you.
Yeah, so it'll be, what do you call that? Translated, and that'll make the first one here, and then the second one here, and then the third one. These proteins are not connected, okay? But they are, what do you say that, translated one after the other. Meaning the ribosome will read, come down this way, and that makes the first one, then it makes that one, the same ribosome, then makes that one, etc. What else did I want to talk about with this? Oh, yeah. In eukaryotes, we make the beta galactosidase, that's simply the lactase enzyme. We make the permease, that's the transport protein that brings the sugar lactose across the cell membrane. And then we make transacetylase, and that's a protein involved in converting beta galactose to the sugar glucose. Okay, we make all of these enzymes or permease. I don't know if that's an enzyme or a transport protein. It's probably both. But we have 3 operons, or you could say 3 lac operons. Prokaryotes only have 1 lac operon. Any question about any of that? All right. Anyways, this is a unique system for turning on a gene. You turn on the gene whenever essentially the sugar is around. And then when that sugar isn't around, you turn off the gene because the products of the gene will not be useful to the cell. And that turns on and off genes. Any question about operons and the expression of gene expression? All right, there's a little movie you can watch on the lac operon. I'm going to move forward because we're running out of time. We're going to then move on to the topic of mutations. Mutation is a change in the genetic material. Mutations may be neutral to the organism, they may be beneficial, or they may be harmful. I think several of you know a mutation in the human population which is harmful. Can anyone mention one? Anyone? Okay, think about that. I'll mention a mutation that is currently neutral, and that is initially 10,000 years ago, humans only had brown eyes or brownish colored eyes. 10,000 years ago in Northern Europe, there was a mutation which created blue eyes. Today, blue eyes and brown eyes, which I don't have a picture of that, those genetic changes are neutral. That may not have been the case in the past because the blue-eyed allele increased in frequency. Either in the past, the blue-eyed allele was beneficial, or it was next to a gene that gave an advantage to the organism, and then the blue-eyed allele got increased in frequency because of its location. Either one. Okay, any question about any of that? But today the blue-eyed and the brown-eyed allele, they're neutral. There's no benefit for one allele over the other. I can give an example of a beneficial mutation too. They saw this when they were studying fruit flies, and the person who was doing the fruit fly lab let the bottle get really full before they changed the bottle, and the pupae in the fruit flies will be on the side of the bottle. And when the bottle was really full, there were no more sites for the pupae to go, so that they had no chance of having, I guess, successful offspring because there's no site for the pupae to go to. And a beneficial mutation happened in the bottle, and it allowed the fruit fly to have the pupa on the cotton at the top of the bottle. And that was just the plug to keep the bottle shut. And that was a beneficial mutation. The mutation then spread throughout the population of fruit flies. Any question about any of that? All right. A mutagen is an agent that increases the chance of a mutation. Now, there are spontaneous mutations, and they occur in the absence of a mutagen. And here we're looking at eye color, and that one would be a mutation. There are different types of mutations. There is a base substitution, which is also called a point mutation. And there's three types of base substitution: a silent mutation, a missense mutation, and a nonsense mutation. And I think you're going to learn about those in the in-class. Nope, I'm going to talk about them later. We have a worksheet where you'll learn about them too. All right, a base mutation is just a single nucleotide which gets switched in the DNA. So something like what might have been ACC gets switched to ACG, a base substitution. A frame shift mutation is a second type of a mutation, and that's where you actually add or remove a nucleotide base. And a frame shift mutation can be adding more than one or subtracting more than one nucleotide. Any question about that? there is an exception, and we'll talk more about the exception when we talk about frame shift mutation in greater detail. The third type of a mutation is a macromutation, and you should listen to this lecture well because your book does not discuss a macromutation. A macromutation is where an entire region of a chromosome is added or deleted. Can anyone mention a macromutation that occurs in the human population? Where an entire chromosome is added. All right, think about that. This is a picture, I read about it. They were studying this boy to see if he was a mutation. It's kind of hard to see the picture there. I blow it up. As you can see, metal spoons are being attached to his skin. And they were wondering, is he a mutation turning into a magneto, if you know what I'm talking about, or is it just a mutation making his skin sticky? And I read that they were studying him, but I never found the paper that discussed what the results were. Was it a mutation or was it just something else? Like, I don't know. He had sticky skin and he didn't ever wash himself or something. I don't know what the study found, so go on. All right, let's talk about a base mutation. A base mutation simply happens when the DNA polymerase reads the template and makes a mistake. So normally the template is T and it put the A and then it has a G here and it makes a mistake and it doesn't put a C here, it puts a T. Now, before DNA replication, this can be corrected, meaning that the cell could recognize the DNA is not pairing here, and it can remove this T and then put in the correct C. And the DNA does have means to repair this if it catches it before the DNA is replicated. However, once the DNA replicates, the G will be replicated correctly, but the T will not be replicated correctly. And now we have a mutation in the population. This cannot be corrected other than a reverse mutation, and we aren't going to talk about a back mutation. I mean, that's a possibility, but the chance of it happening is very slim. And to tell you the truth, the chance of this happening is very slim as well, meaning the first mutation. And in this mutation, let's see, TGT is correct. So it made the amino acid cysteine. And you'll see that this one here, let's see, it was supposed to be TGT, and now it's TGTACA. What's wrong here? Oh, sorry, I'm missing where the mutation happened. So it's from the T, and it made initially the amino acid cysteine, and now it's making the amino acid tyrosine. So this is changing the not only the DNA, but also the protein. Any question about any of that? Excuse me, I had to blow my nose here. All right, so let's talk about a silent mutation. A silent mutation is where there is a change in the nucleotide basis of the DNA, but the protein is not changed. For example, the amino acid UUU codes for the protein phenylalanine. If there is a base change and then the codon is UUC, because the mutation, it does not change the protein because the same amino acid phenylalanine is coded for. And this is a silent mutation where the DNA is changed, but the protein is not changed. So there's no change in the protein, no effect on the protein function either. Any question about any of that? There can also be a missense mutation where one nucleotide in the DNA is changed and it results in a change in an amino acid in the protein. For example, the DNA was CCG, coding for the amino acid glycine, and this DNA gets changed, mutated. Am I reading that wrong? CCG. Oh, here it is. The C got changed to a T, and then that's the CG. So TCG changes the amino acid glycine to the amino acid serine. That results in a change in the protein. The protein will probably fold differently, and the protein will have probably less activity, meaning it won't work as well, or it may not work at all. That would generally be a negative mutation. If the protein does happen to have the same folding pattern, then it would be a changed protein, but the protein would still function. Any question about any of that? Anyways, that's a missense mutation where the DNA is changed and the amino acid is also changed in the protein. There's also a nonsense mutation where the amino acid, the nucleotide is changed by one nucleotide, and it changes the protein. Instead of coding for the amino acid glycine, so that was GGC, well, it looks like this one was changed. Yeah, this one was changed. So instead of coding for the amino acid lysine, AAG, it now codes for UAG, which codes for a stop codon in the protein translation. This will create a truncated protein. The protein will get made up to the stop codon, but then the protein is truncated. The rest of the protein isn't there. Oftentimes, this protein will not have any function. Sometimes the protein has reduced function. I've never heard of a case where it happened and the protein had normal function. So usually this is one where the protein will have reduced or no function. A stop codon. Changing it from a base substitution to a nonsense mutation. Any question about any of that? All right. A frame shift mutation is where we get an added nucleotide to the DNA, or where one base gets removed. So instead of having 3 A's, one base was removed. Now we only have 2 A's. What happens if a frame shift mutation is when the ribosome is reading the nucleotides, it'll still read the codon as 3. So instead of reading this as, come on, mouse, instead of reading this as A, because the A is removed, it'll now take the next codon or the next nucleotide and add it to this codon. So it'll be AAC, and that will change this protein, the amino acid in the protein. But it also changes all of the codons after this frame shift mutation. So instead of going, what is it, CCG, it'll now go CGA. So it'll start here at the start of the codon, and then the next codon, and then it takes the nucleotide from what used to be the next codon down to make this codon. And so the protein will be changed from the frameshift mutation. Any question about any of that? Oftentimes, this will make the protein less active or having no activity. That happens whether you remove the nucleotide in a frameshift or you add one. If you add an A here, for example, this amino acid will be correct, but then the next one will be wrong because instead of going CCG, it'll be ACC. There is one frameshift mutation which is not considered a frameshift, and that is when you add or subtract a multiple of 3. For example, if we were to remove all 3 of these A's, we will get the same protein. It's just it'll be missing this. Come on, mouse, dang it. It'll be missing this one amino acid in the protein. And if you get a multiple of 3, either added or removed, it's not called the frameshift mutation because the frame of the codons is not shifted. Any question about any of that? All right. When we're talking about mutations, there can be 2 types of mutations. There can be spontaneous mutations, and they just happen all the time, actually, not very often, but all the time, simply by the DNA polymerase making a mistake. And then you can have a mutagen, which also causes the mutation. And there are 2 types of mutagens. There's radiation, and then there's chemical mutagens. Both of these alter the DNA, and they increase the chance of the mutation happening. Spontaneous mutation, do I have that here? I'm sure I do someplace. Ah, there it is. So we'll wait on that. So remember that there are 2 causes of mutation. There can be a spontaneous mutation, and then there's mutagens, which will increase the mutation rate. There's 2 types of ionizing radiation, or I shouldn't say ionizing. There's 2 types of radiation. There is ionizing radiation, and that is radiation with strong energy like X-rays and gamma rays. And when that energy hits the atom, it'll actually give the electron enough energy for that electron to absorb the energy and then be ejected from the atom. That will make an ion because the atom is losing an electron, a negative charge. And then this ion can then react with DNA, mutating the DNA. Any question about ionizing radiation? There's also mutation, which is radiation, which is different, and that's non-ionizing radiation. And most of us call this UV light or ultraviolet light. UV radiation damages the DNA. Let me blow this up. When UV radiation hits DNA, it damages 2 thymines which are next to each other, creating the thymine dimer. This is actually a covalent linkage between these 2 thymines. these thymines will now no longer hydrogen bond to the A on the corresponding strand of the double-stranded DNA. That'll cause a kink in the DNA right here. Okay, we're not going to talk about it, but the light repair mechanism can actually remove the thymine dimer and correct that. So the cell does have means to correct this. But whenever you get exposed to UV light, like your skin and your sunbathing, you create thymine dimers. This stops both DNA replication right here at the thymine dimer and the RNA polymerase, which is trying to transcribe the gene. Now let's talk about the third case of a mutagen, and that's a chemical mutagen. We're going to give it as an example of nitrous acid as the mutagen. Nitrous acid, when it comes into contact with DNA, can change the adenine so that this adenine is chemically changed, but it is adenine, but it no longer will bind to T. And when the DNA polymerase sees this changed A here, so it reads this A, it's not going to read it as an A, it's going to read it as a G, and then it'll insert a C across from this changed A. Come on, mouse. And then when the DNA replicates, this C will code for a G, and now this mutation is permanent. The cell can no longer correct that. Okay. I'm not sure how the cell would repair this, but it is possible that the cell could recognize this A is altered and then remove it and then put in the correct A. That would have to happen fairly early because it could read this one and know to put in an A here. Right here, it would have a hard time correcting that. All right, any questions about how nitrous acid mutates the DNA? So let's talk a little bit about the frequency of mutation. Spontaneous mutation, where the DNA polymerase just makes a mistake and adds the wrong base, happens for one base in 10 to the 9th replicated bases. So a spontaneous mutation occurs in 1 times 10 to the 9th replicated bases. Now, for each gene has about 1,000 bases. So that means that one gene in 10 to the 6th replicated genes will be mutated. What is the population of the human population on Earth? Come on, somebody's got to know that. Is it 1 million?
Is it like 7 billion?
Yeah, it's 7 billion. So when children are born, and we're only looking at one gene in the entire human population, we're clearly going to get a mutation every year in the human population in this one gene we're looking at. You guys all see that. And that's because the human population is about 7 billion, going on almost 8 billion. And so we'll always have mutation happening in the human population because there's so many children being born. And I don't know what the birth rate is. But it's certainly more than 1 million children born in a year. Okay? So we'll have a mutation in the human population every year. And we're only looking at one gene here. Any question about any of that? Anyways, that's the frequency of a spontaneous mutation. Mutagens increase the mutation rate 10 to 1,000 times higher, meaning instead of one gene in 1 million genes being mutated, it'll be one gene in 100,000 genes, or 10 to the 5th, or even one gene in 1,000 genes. So mutagens really do increase the frequency of a mutation. What's the most common mutagen that humans are exposed to besides sunlight? Nobody knows?
Chemicals,
Say again?
chemicals in the air.
No, that would be not a common mutagen. I mean, we do get exposed to chemicals at low risk, but the mutation rate is not that common. It's actually cigarette smoke. Lots of people smoke and they're being exposed to the mutagens in cigarette smoke, and cigarette smoke is a really strong mutagen too. There are different ways we can identify a mutant experimentally. You can do positive or direct selection, and then there's negative or indirect selection. Positive direct selection is where you detect the mutant cell or the mutant organism because they grow or appear differently. Like if this individual was ever walking down the street, people would say, yep, there goes a mutation. Right? Negative or indirect selection is where you detect the mutant cells because they do not grow. Let me discuss negative selection of mutations a little more detail so it's clear to you. I used to do this when I was actually in college, and that is you can take a replica plate of colonies on a plate. So you'd make the colonies separated and isolated on the plate, and then you could get a velvet stamper. It has velvet like that, and you stamp the plate. Now the cells from these colonies are put into the velvet. So this colony, which one are we interested in? That one there, which looks like that one there, is put right there. And then you stamp 2 plates, one with media containing the amino acid histidine, and then one with media lacking the amino acid histidine. And then you allow the cells to grow up. If you notice that there's a colony growing here, which is this one here, and it's not growing on the plate with media lacking the amino acid histidine, you can say that these cells are a mutant and they require the amino acid histidine in order to grow. So this is negative selection for mutations. And I used to do this looking at bacteria, trying to find cells that were mutants. Any question about any of that? I actually did that as an undergraduate. I don't remember doing that as a graduate. All right, let's move on to genetic recombination. Recombination is the exchange of genes between 2 DNA molecules. It's similar to crossing over. Crossing over is where 2 chromosomes, the mother's and the fathers get together, and then they literally cross over, and then they break right here where the mother's was blue chromosome. And now we have the mother's chromosome to here with the capital letters, and then the father's chromosome here in the white color and the lowercase letters. So crossing over happens in mitosis, actually mostly in meiosis, between the mother's chromosome and the father's chromosome. And this would happen in women if both of these were X's. It doesn't happen if it's an X and a Y because they don't actually cross over in X and Y because they're different chromosomes. But if this is the first chromosome from the father and that's the first chromosome from the mother, they pair together and then they can cross over. Crossing over is where the chromosomes pair, cross over, and then break, where each chromosome is then a combination of this one being the mother, the blue, and then part of the father's, the white. Recombination is a little more general, and that's just an exchange of genes between two DNA molecules. It may happen with crossing over, but it can happen in other cases, like we'll talk about transposons in a little bit, where one gene can move from one spot to another spot, and that would be recombination, exchange of genes between two DNA molecules. But generally they are the same, recombination and crossing over. It's just recombination's a little broader term. Any question about any of that? All right, let's talk about genetic transfer. There's vertical gene transfer and horizontal gene transfer. Vertical gene transfer occurs during reproduction between generations of cell, from the parent cell to the offspring or daughter cells, or for humans from two parents to one offspring. Horizontal gene transfer is the transfer of genes between cells of the same generation. So a donor cell sends DNA to a recipient cell. Horizontal gene transfer can happen in 3 different ways. It can happen with transformation, with conjugation, and with transduction. Let's talk about each of these. Transformation is where naked DNA comes out of a cell. Usually, that cell dies. And then we have naked DNA in the environment. The naked DNA comes to the recipient cell, and then the recipient cell brings in that naked DNA. And then part of that naked DNA can recombine with the chromosomes and then be expressed by the chromosome. So of the naked DNA being brought into the cell, only the lowercase a recombines with the host chromosome, and then the lowercase a is added to the genome of the cell. The rest of the naked DNA is degraded and then can be recycled as nucleotides for the cell. You don't need to know that, but the A recombines with the host chromosome, the lowercase a, recombines with host chromosome, and then that lowercase a can be expressed, giving the cell a new gene, the lowercase a gene. Any question about any of that? Transformation is one way that a cell can gain new genetic information. This lowercase a gene was brought into this cell and then expressed by this cell by the process of transformation. Any questions about that? Here we have a little picture of transformation where the cell brings up naked DNA, then digests that DNA and then expresses it. And the cell phenotype can change from the newly brought in DNA. And that's the process of transformation. Any questions about any of that? Transformation is frequently the way that molecular biologists add DNA in the lab to cells, like the flavor saver tomato, if you remember hearing about that, or most of the genetically modified crops, they were made by transformation. Transformation can occur in the wild as well as in the lab, but this is generally the way molecular biologists add new DNA to a cell. A second way that a cell can get new DNA is with conjugation, sometimes called sexual conjugation. There are two ways that conjugation can happen. We're only going to talk about it in the case of gram-negative cells. Gram-negative cells have a genetic makeup where there's actually a F plasmid in this cell. Oops, sorry, I got the wrong one. In this cell here, this is the F plus cell. If I could read that, I could do this. Let me see if I can blow this up a little. There, that's better. Now I can read that. That's the F plus cell. It has an F plasmid in it. The F plasmid codes for the production of a sex pilus. The sex pilus goes out and literally will skewer an F minus cell. And then it can bring the two cells closer together. They may be brought touching, meaning it acts like a winch and actually brings the two cells, the F plus cell and the F minus cell, closer together. However, it doesn't have to bring the cells that close together because the sex pilus is hollow, and that will allow the F plasmid to replicate itself and then send a copy of the F plasmid into the F minus cell. And that we're seeing here where the plasma is being replicated and then brought into this cell. And now this cell has the F plus plasmid. And this cell will code for, or the plasmid will code for the sex pilus, and that will change the phenotype of this cell from an F minus cell to an F plus cell. And then this cell can then go out and skewer F minus cells and then transfer the plasmid into the F minus cell, converting that F minus cell to an F plus cell. Any questions about any of that? That's what happens in the gram-negative cells, that they have this F plasmid, which allows sexual conjugation to happen in this manner. For the F plus cells, they can also engage in sexual conjugation. The F plus cells do not have a plasmid coding for a sex pilus. What they do is the two cells come together and then touch. And then the two cells touch and the cell membranes touch, and then the cell membrane actually opens up so that the cell membrane, instead of going around this way and around this way, the cell membrane of this cell comes around and then combines with the cell membrane of this cell and then goes around the cell this way and then this way. That can allow the cytoplasm from this cell to move into that cell or vice versa. That can allow a plasmid in this cell to move into this cell or vice versa. And that is how sexual conjugation can happen in a gram-positive cell, where the two cells touch and then form a mating bridge. And the cytoplasm of this cell is connected to the cytoplasm of that cell. Any questions about sexual conjugation? In sexual conjugation, we always have DNA moving from one cell to another cell, and that DNA is usually a plasmid. You can also have transduction, move genetic information from one cell to another. And I forgot to mention that here. Well, I sort of did mention it here. And that is the gene here on the F plasmid move into this cell. So this cell gets new genes. And then that changes the phenotype of this cell. So let's talk about transduction. The third way that horizontal gene transfer can happen, where DNA moves from one cell into another cell. Transduction happens with a virus, with a bacteria virus, which is called the bacteriophage, or just phage for short. What happens is the viral genome is injected into the host cell. And then the viral genome, what it does is, the first thing it does is it degrades the host cell's DNA. The reason it does that is the viral genome wants to determine what's going to happen in the cell. And an easy way to do that is to degrade the host chromosome. The viral genome then takes control of this cell and then replicates itself, making more viral genome, and then making viral protein to make the virus. And then the last thing the virus does is it lyses the host cell. And when it does that, it'll release the newly replicated viruses. When these viruses go on and infect another cell, we'll follow the same procedure as this one. However, let's see if I can blow this up a little. You'll note that the DNA of the host chromosome is purple. The genome, we'll just say it's DNA of the virus, is a reddish purple. So bluish purple, reddish purple. Viruses occasionally make a mistake where instead of stuffing the viral genome into the virus, it actually stuffs a piece of the chromosome for the host cell into the virus. So that's the dark blue purple DNA put into this virus, which is the DNA of this cell. When this virus now goes on and infects this cell, it will inject the DNA from this cell into this cell. So this recipient cell will now get new genes, the genes of this cell. And this DNA can be incorporated into the chromosome, and then that donor DNA from this cell can then be expressed. So this we call the recombinant cell, the cell that got the new DNA from this cell. Any question about any of that? All right. So those are three ways that horizontal gene transfer can happen: transformation, conjugation, and transduction. How frequently do transformation, conjugation, and transduction happen? It's about 1 times in every 10 to the 5th genes. So transformation, conjugation, and transduction happen about 10 times more frequently than spontaneous mutation. So oftentimes, if a cell is expressing a new gene, it will be from either transformation, conjugation, or transduction. And it'll be 10 times more likely from these three than by spontaneous mutation. You might think about that if ever you're working in the hospital and then you have a patient which is resistant to antibiotic resistance. It's true that antibiotic resistance could have happened by mutation, but it's also true that if that mutation already existed in the population, the cells in that patient may have gotten the resistance, the antibiotic resistance gene, by transformation, conjugation, and transduction. And the frequency of these are 10 times more common than the frequency of a spontaneous mutation. Any question about any of that? Transduction probably happens in human nose cells all the time, but that doesn't change the human germline, so nobody cares about it or follows it. But conjugation and transformation do not happen in the human genome. Okay? Any question about any of that? Or I should say, they do not happen normally in the human genome. Obviously, in the lab, something could happen because the scientist is making it happen. All right, let's turn to plasmids. Plasmids are a small circular extrachromosomal DNA. And being circular, they take on the same form as the chromosome of a prokaryotic cell, meaning circular chromosome. However, the difference between the chromosome and a plasmid is one of size. The plasmid only has a few genes on it. The chromosome has thousands or maybe even hundreds of thousands of genes on it. Any question about any of that? So here we're looking at one plasmid, and there's a gene there, and a gene there, and a gene there, and maybe one there. I'd have to blow it up, one here. So that's just a few genes on this plasmid. Plasmids do replicate independently from the cell's chromosome. And we actually saw that right here, where this plasmid replicated and the chromosome did not. Because the cell didn't go through cell division here, only the plasmid replicated. And then one copy went into that cell. Any question about any of that? Plasmids are sometimes seen in prokaryotes. Eukaryotes, most eukaryotes do not have plasmids. There is an exception though. Yeast cells do have plasmids. So in humans, all of the DNA is either in the chromosomes in the nucleus, or the DNA is in a mitochondria or chloroplast. And if I said humans, I didn't mean chloroplast are in humans, and eukaryotes. But yeast do have plasmids, and then bacteria have plasmids. You should know that there are different categories of plasmids, but I'm not going to ask you to learn the different category names. Just know that there are different types of plasmids. And then there are transposons and insertion sequences that come along with the transposon. What a transposon is a piece of DNA that jumps around in the genome. So a transposon really is a jumping gene, and it has an inverted repeat on both sides of the transposon, and at the minimum, it encodes the transposase gene. Let me blow that up. The transposase gene, so let's say this is a chromosome here, and the transposon is right here in the middle of the chromosome. The transposase gene codes an enzyme which cuts out the transposon right here and right here, and then it anneals the DNA together because you don't want your DNA cut apart. And then the transposase gene goes to another location. It could be another chromosome, and then cuts that chromosome and then inserts itself into that cut chromosome and then anneals the chromosome. That's what a transposon does. It's a piece of DNA that moves from one place to another place in the genome. In eukaryotes, a transposon can move from one chromosome to another chromosome. Complex transposons also carry other genes. Like this transposon TN5 has the transposase gene, meaning IS1 here on this side and the same one here. So in reality, it has 1, 2, 3, 4 inverted repeats. The important one is this one here and that one there. And then it carries a gene in the middle of it, which is the gene for kanamycin resistance. And then that can spread the kanamycin. Well, this gene moves from one location to another. So it could be in a place where this gene was turned off and then moves to a place where this gene is turned on. And because it is a transposon, it can move from the chromosome and then move into a plasmid. And then the plasmid can leave that cell and then go into a recombinant cell, bringing with it the kanamycin resistance gene to that new cell. And that's how transposons can move genetic information around, not only within the genome, but between cells also. And if moving between cells, realize the transposon has to move into a plasmid, and then the plasmid has to move from one cell to another cell. All right, any questions about any of that? If not, let's talk about our last slide. Oops, second to the last slide. This is showing you the transposon, which moved from the chromosome to a plasmid, and it brought with it the kanamycin resistance gene. Now let's go to the last slide. Genes and evolution. Genetic variation, if it is new in the population, the only way it can arise is by mutation. But once the genetic variation is in the population, genetic variation can result from, yes, mutation, from transformation, spreading from one cell to another, conjugation and transduction, and then transposition. Crossing over doesn't really spread the genetic variation. It just reshuffles the genetic information, putting new genes together. Like where one gene that was on the father's chromosome moves to the mother's chromosome, and now that gene from the father's chromosome is paired with a gene on the mother's chromosome. So crossing over is important in genetic variation. Like I said, it doesn't create the genetic variation, but it creates new combinations of genes. And then this genetic variation drives evolution. And evolution will proceed by natural selection. So natural selection will work on the genetic variation, increase the frequency of good genes, decrease the frequency of bad genes, and then that will cause the population to evolve. Any question about any of that? Any question about any of chapter 8? Okay, if there's no questions, I'm done. I'll see you in the lab at 6:30.